Don't switch me on. No worries, I'm good. Okay. Okay, good. So let's get started. So thanks for the kind introduction. Thanks for having me. It's really nice to see so many familiar faces again. And also some people who joined our workshops that Marcus and I held in the past last year and before the pandemic. So still they're working with Optimus. It's very nice to see that we didn't frustrate them too much during this courses. So what I'm going to talk to you about is very briefly the things that we're doing, and then we go into a specific project that we have developed over the past 8 years or so to give you more details about stuff. And as you know, mostly I try to talk about work that we haven't published yet. So just to share something that we are quite excited about. So our lab is currently doing a lot of work with optorabogenetics. So using aptamers together with photo switches to generate genetic elements that can respond to light, and there we can switch on and off. And gene expression. Teja from my lab is here. She has a poster. So please go there, talk to her. She knows everything about this. And we published some of the work is not published yet. So just take the chance and talk to her. I don't have to say anything about this part. So we're doing cell selects, complex target selects, and all those things since years, since 20 years almost. For the purposes we have just seen here, to find aptamers that can target cells that can internalize, but also as a discovery tool to find targets on the cell surfaces to surfaceome analysis, because we still think that aptamers can do something that antibodies cannot, and is binding complexes that targets in a way that we can find in a native environment. So I'm not talking about this today, and also not talking about this. So we have invested quite a lot of time to develop this quick helix methodology, published a little bit on it, and founded a company which has been sold lately to a company in the US and the UK. And Marin is here. She founded the company with us. She was also at the oligo meeting. So talk to her if you want to know anything about this. Just that I can talk about this today. So we have spent 8 years or so from now to develop aptamers that bind to chemokines and use them in vivo to target them to reduce contact allergy responses. And this work has been inspired and started by a colleague of mine, Irmgard Förster at the Limes Institute, the same institute where I'm hosted. And she's working with these chemokines CCL17 and CCL22, and found out that they are upregulated in contact dermatitis. So we thought maybe it's worthwhile to do some aptamers and see where we can reduce this, what we can learn here. So the work that I'm mainly presenting is the work that has been done by Anna. She's also here. She also has a poster on this topic, and the work from Marlene. She's a PhD student in Irmgard's lab. So they work very closely together. Marlene mostly doing the animal and cell culture work, and Anna did all the work with the aptamer selecting and characterization. Okay, so CCL17 and CCL22, they're upregulated in many diseases, especially contact dermatitis. So upon exposure to an antigen, what happens is there are two phases. So I literally know next to nothing to this, but I try to put it in my own words that everybody can understand it. So what happens is upon the first exposure to an allergen, not really much happens on your skin, but it happens a lot in the body and with the T-cell responses. So there are dendritic cells, macrophages that take up those allergens. They are then dissected and presented on the cell surface, and then they induce T-cell responses. And then mainly in regard of memory T-cells and regulatory T-cells, but on a phenotype level, nothing serious happens. You don't feel it. But the worst thing happens then after the second exposure. After the second exposure, we have the T-cells that remember that there has been this allergen contact before. If you have the contact with the allergen again, then there are these chemokines among other signals that will be upregulated, at the point where everything happens. And then the T-cells are migrating to the site of action, and you get the skin rash and everything, and cytokine storms, you would say. Okay. So that's basically what can be done. So for CCL17, we have identified 2'-fluoropyrimidine modified RNA aptamers a couple of years ago. We also published on them. And what we, after developing them, we truncated them a bit, we pegylated them in a 5' end, that cap structure, and 3' end that's not shown here. And then we subjected them to a mouse model. So they're mouse models for contact hypersensitivity. And they're mimicking just what I've said in the slides before. So you have a sensitization phase in which you expose the animal on the belly with a model antigen. This is an DNFB, so dinitrofluorobenzene. Then you have the sensitization phase. This happens 5 days prior to the challenge. So this is the second exposure then. Then you do the second exposure specifically on the ear with the same antigen. And then you measure the swelling of the ear after 3 days of exposure. So if the ear swells, then there's this allergic response. If the ear doesn't swell, there's something that inhibits it. And what we did is we, on the challenge, we injected directly the aptamer into peritoneum. And after 12 hours, we again injected it into peritoneum. And then we measured the ear thickness. So then for the CCL17 aptamers, you can see that, okay, both aptamers somehow diminished the swelling of the ear, and very nearly to the knockout of CCL17 is a genetic knockout. We still have some ear swelling, but less as the vital elements that haven't been treated with any of them. Okay, so far so good. That work is published. So what Irmgard also found out is that if you knock out CCL22, as mentioned before, it's also excreted on the site of allergen exposure. And if you knock it out, also there, the ear swelling is diminished. So logically, next step was, can we identify aptamers to this compound as well, to this protein, and that can reduce the ear swelling too. So that's the work that Anna has started. We have to say that she has done tons of selections. So with 2'-fluoromodified RNA, didn't get anything useful. We used manual selections, automated selections, DNA, and so on and so forth. In the end, what worked was a DNA selects. So plain DNA, no pigments, nothing, it's just DNA. That's a typical analysis that we do, so binding analysis of the enriched libraries, sequencing of millions of candidates for each selection cycle, diversity analysis. So we see here that's not really very much enriched, so still have a level of around about 25 to 30% of unique sequences in the around 10 library, which is still a lot. But you see that most of them, so 70% of them are enriched in a way that they have gained a lot of copy numbers. So Anna did a lot of analysis and so on and so forth. So she chose 9 sequences here called HA1, 4, 21, and so on to HA104. Did initial binding assessment. So this is a non-binder, all the others need to bind. And with this, we went further. So the next step was, there's a model, and you can do in cell cultures, you use something like a transwell assay. So there's a well, and then you have a cap inside. So you have a lower compartment and upper compartment. In the upper compartment, you put the T cells that react on the chemokine. Then in the lower compartment, you add the chemokine. And this is a cell mechanical membrane, so that the cells only migrate through this membrane if they feel the presence of the chemokine. And if you have this assay up and running, you can add the aptamer in the down, up in the lower compartment and see whether it can then interfere with the movement of the cells. So this, and in an ideal world, the experiment result would look like this. So I show you this example because the other side will be very busy, and we go through it very quickly. So we have in the aptamer the CCL22. So this is the migration of the cells in the absence of aptamer and the absence of the chemokine. Then you have the presence of the chemokine only. That's then our normalization value. So that's the maximum migration you would detect. And then you add a control. And I'm saying this, so what we usually do in our lab is, at the first place, we have an aptamer we'd like to investigate. So we're generating a control, and that's a scrambled control. So the scrambled means parameter the same, but the selected sequence has been scrambled, same sequence composition, but in a different order. Later, we go for point mutants when we have more idea about structure and so on. But at that stage, you don't have this. that for every aptamer we investigate, we are generating these controls. So that's the control. So you see that does not inhibit very much. And if you have increasing concentrations of the aptamers, we see a strong inhibition of migration. And these +++ means, so here we have a ratio of chemokine to aptamer, which is 0.1. Here it's 1,1, and it's tenfold excess of the aptamer, so just as an average amount. Okay, so that's a busy slide, but in the end, so what you can see from this slide, or maybe I should point this out, that some of these controls that we designed, like here, for instance, are also inhibited. Yeah? So it's something we see sometimes, that the scrambled have some effect. We don't know why, but we see this, and this becomes a bit more important later on. But based on all these data, we choose HA82 and HA102 to further characterize them in the subsequent data, subsequent project analysis. So what we then usually do is something that happens in parallel, more or less. So we do truncation, binding affinity determination, and so on and so forth, specificity. So that's why these data are not usually in the chronological order how I show it to you. It's just like that, that's the final result you obtained, and I think the way we show it here would make sense. So what Anna did is she truncated both aptamers. For 82 aptamer, we could chop it down to 37, 51 sequences. What you can see here, that's the full-length sequence with primer. That's the truncated one, so these grayish represented sequence parts, they are they're not present, they're absent. And we introduce a point notation which should close this putative stem. So these M-fold predictions for DNA aptamers are usually very shitty, I would say, so it's not working very nicely, but it's nice to show at least a bit of structure. But for DNA aptamers, my impression is they don't work well. So for the 102, we could chop it down even further. So the 29 worm, just shown here, works still very nicely, binds well, and these are actually two sequences, the 51 from 82 and the 29 from 102. They were then pursued further. So we did some specificity analysis, and the aptamers are very specific, as you can see here. Unfortunately, they only bind to the murine versions of the chemokines, so urine but not human. But it's the same thing that we have observed for the CCL17 aptamers. They only bind to the murine but not to the human. So we need to do extra selects to target those. So under the binding determination, that the curves you see are binding for flow cytometry. So we measured flow cytometry data. Full length truncated versions are always a bit better, maybe because of sterical issues. But we did also SPR. Also, this SPR in this case gave better results. We often get different KD values when we measure with different techniques. So it's nothing we're concerned about, but just happens. So, but the affinities are fine to go further. Now, I think that I'm showing this, it's busy, but I walked you through, but I think it's important, especially in this audience. So the process of bringing these aptamers from in vitro to in vivo will be that we are doing characterization in vitro, then we truncate, then we do for applications in vivo pegylation and a cap DT at the 3' end. So what happens here is the following. So these are the migration assays, transfers that I just showed you. So this is the data set that you have already seen. So all the controls, the scrambled versions into different concentrations, and the aptamer. So far so good. If you now, so this is the full length. If you now use the truncated version, still it has a nice inhibition of migration. But if you use the corresponding truncated version of the control, see that it starts also inhibiting, which is kind of a surprise because we did not expect this. So we had to reassign new controls. That's the control B here, that then does not have a strong effect, not on a naked aptamer without modification, and not on a modified one with the pack and the 3' cap. For the 82 aptamer, the situation was surprisingly the same. The truncated control also showed kind of inhibition, but still significant. So we had to change it, and we changed it for this control B then. And this here, it says it's at high concentration significant, but we nevertheless went on with this. But it shows us that although we have designed a control in the very beginning, we cannot just assume that if we modify it to some extent, we can use it just right away. Because if we would have done this, we would have seen probably very different effects in vivo. So what we also do is prior to, well, in this case, we did it prior, we did it after, but prior to injecting those aptamers into the mouse, we do a test whether they induce innate immune responses. And so far, we always get kind of sometimes sequences that can do this, that initiate innate immune responses. So you treat macrophages with the aptamers, and you measure TNF-alpha release. And that was actually the case here. So for the 82 aptamer, we see a very, very strong innate immune response. Whereas for the other aptamer, we don't see it. So that's the naked aptamer. And this is the pegylated and capped aptamer versions. Same here, a little bit less for the 82 aptamer, but the other one, they're both very clean. Also, the controls are clean, which is important because otherwise we cannot inject them really, because we would evoke different immune responses. Fortunately, our very first data that we did with mouse showed that 82 doesn't work in the mouse. But what we could show is that the 102 works in the mouse. So the same treatment regimen, sensitization, then the challenge injection of the aptamer, at the same time of the challenge, and 12 hours later, so we inject 10 nanomoles, and then we measure the ear thickness after at day 0, day 1, day 2, and day 3. So that's the knockout, that's the activity of the aptamer, that's the control aptamer, that's the white-haired mouse. And these are the non-challenged animals. So we see that there is a reduction. So what we then did, or Marlene and Anna did, they said, okay, well, we can use maybe the aptamer just as a cream on the ear. What they investigated, so this is a so-called assay chamber. So you use a skin, a sample, put it into the chamber, so like here, and then you close it, you put aptamers up here, and then you wait for a while, and then you take it out, and we did this with fluorescent labeled aptamers, and then you fluorescent microscopy to see where the aptamer goes. So that's the cream only, that's DAPI, that's the aptamer stain that was labeled with a red dye 647N, I would say. I hope that's true, but it's in the reddish area. And that's the aptamer signal we got. And you can see, if you zoom in, that it's not only here in the epidermis, but it's also in the dermis. So it actually goes in, right, somehow. Yeah? We don't know why, we don't know how far, we don't know what is the kinetic, we have no idea what is the stability, and so on and so forth, but we know that it can inhibit. Yeah. So they did the experiment, again, sensitization, then the challenge, along with the cream on the ear, and then after 12 hours, another batch of the cream on the ear, and then measuring the thickness. And the results are here. So this is in green, the knockout. That's the aptamer. That's the cream with the control aptamer, and that's the white-type mouse. Yeah. So it seems to work, kind of. So that's just, I think, very promising. And so that's where we are with this project right now. We're about to wrap up the data in a manuscript and try to publish it. We're doing a few control experiments, but I think it's mature enough to show it here and to share this experiment. So that's a summary. I don't repeat what I'm just saying. So, but what we're doing and what Anna already did is she's trying to select and selected aptamers for the human versions. So there are different ways how to do it. You can do it just for the human chemokines. You can do toggle settings and all these things. So we now choose just to do the human counterparts and see what we get there, just to move on, because with this we can do proof of concept, but we cannot move into patients. So this brings me to the acknowledgement. So all the work I'm showing was from Anna and from Marlene. So those two work really very closely together. It's an excellent project. The innate immune response were done by Michael Eikelatz's group. Matthew there. That's not my group, but that's the entire chemical biology unit of the Linus Institute at the barbecue last year. We are searching for persons to join us. We have PhD positions and postdocs available. So if you know somebody who's interested, just tell them. The entire work was sponsored by the German Research Foundation, and we're having the workshop again this year in Bonn. So spread the word, and I thank you for your attention.